Hello there all. Welcome to another session of ISGB RSG-Türkiye webinar sessions. Our webinar is going to start now. So here are our social media accounts. You can join us and also follow us from the various social media accounts. And we would like to welcome you if you would like to join and work together work with us. And so today's presenter is Associated Professor Dr. Ozge Kurkcuoglu from Istanbul Technical University. The topic of the presentation is using residue interaction network model to identify putative allosteric sites on protein structures. So the stage is yours, dear Ozge hocam.
Yeah, thank you, Deniz. So let me just share my screen. Yeah, somehow my is not okay. Let me try to share my entire screen because I cannot see my yeah. Okay. So okay, I hope that you are able to see it. Yeah, thank you. First, I would like to thank the RSG-Türkiye for this invitation. I'm very excited to this opportunity to talk about our studies using the residue interaction network models to identify putative allosteric sites on different protein structures. So what I plan today is to talk about our results on GPCRs, and also I will be sharing our results with the bacterial ribosome as well as the glycolytic enzymes. So in all of these protein structures, we used the residue interaction network model along with other techniques. So the majority of approved drugs target proteins, and understanding the structure and dynamics of the proteins is very important to develop new effective therapeutics. And proteins are essentially biopolymers that have a unique three-dimensional structure and topology, which enable them to make certain motions to fulfill their biological functions, such as the lead closing of this adenylate kinase over the catalytic site. And these motions are maintained by the three-dimensional structure, which are actually the three-dimensional structure is maintained by the non-bonded interactions such as hydrogen bonds, disulfide bridges, electrostatic interactions, etc. And so far, many physical models have been proposed to describe the protein structure and to understand their functional dynamics. And one good strategy to identify new drug binding sites on a protein complex is to reveal its allosteric mechanism. And allostery means regulation at a distance. Ligand binding to an allosteric site can have an inhibitory effect on the protein activity by shaping the orthosteric site from a distance. And an opposite effect is also possible with an allosteric activator. The current view accepts all proteins as inherently allosteric, and now more and more evidence indicate that there exist signaling pathways that just transmits the perturbation at an allosteric site to the orthosteric site using tertiary interactions. Currently, this regulation at a distance is used to design allosteric drugs, which can have high specificity as well as low side effects, given that the orthosteric sites are highly conserved. And once undruggable targets may now be targeted using allostery. One important struggle in using the allosteric sites is drug-resistant mutations, but there exist many strategies to overcome this problem, such as prescribing drug cocktails, targeting parallel pathways, or designing bitropic ligands, as well as using different warheads in the allosteric ligands. And so far, Numerous computational methods have been developed to reveal the allosteric regions and allosteric pathways. And here I would like to present a computationally efficient method to investigate these using the contact topology of the protein complex. It can be applied to any system involving amino acids, nucleotides, or crystal water, or cofactors. We start by the three-dimensional structure of the protein, either taking from experimental data or some models, such as from molecular dynamics simulations. And then we describe the structure as a network of nodes linked by edges. Nodes are placed on alpha carbons or phosphor atoms, if there are any nucleotides present in the structure. And the neighboring nodes within a 4.5 angstrom are linked with each other, and it becomes a bidirectional weighted graph. And the weight of an edge is calculated based on the local interaction strength. And here we assume that a higher number of interactions brings nodes closer. So that's why we take the inverse as a simple model. And in addition, taking the inverse of the local interaction strength in the assignment of the edge length, remove the strong bias towards the bonded interactions over the non-bonded interactions. And then we calculate the centrality measures of the nodes forming the network. And among these, we have the degree, closeness, and betweenness scores. Degree is simply the number of neighbors of a node, and the closeness shows the topological effect of one node over the other nodes. And we are particularly interested in the nodes with high betweenness values, and because these are the nodes that are frequently visited by the shortest paths spanning the entire network. So they have a high capacity to send and receive an information throughout the network and between distant sites. So in our calculations, we just construct the distribution of the between scores, take the top 5% values, and just look at the nodes with these values and consider these as the hub residues. So that's the name that we give. And so far, we investigated many proteins with this model, such as the G protein coupled receptors, or briefly GPCRs. GPCRs constitute the largest superfamily of cell surface membrane receptors. They are encoded by approximately 1,000 genes, and they share a conserved 7 transmembrane helices connected by intracellular and extracellular loops, which, by the way, are highly flexible for functional reasons. And they are at the center of the signal transduction vital for the cellular activities, and are triggered by a variety of ligands, such as ions, photons, neurotransmitters, hormones, peptides, etc. And the ligand binding from the extracellular site, the receptor transmits the signal allosterically to the intracellular site to start the biological response. And these receptors are implicated in numerous diseases and therefore are considered as targets in drug development studies. And the majority of a high proportion of the currently approved drugs actually targeting the GPCRs. But still, they have highly conserved orthosteric sites, which bring the issue of the side effects. So we can use allosteric, if we just understand the allosteric mechanisms or review their allosteric size, so we can able to develop more efficient drugs with fewer side effects. Here, to assess the ability of the residue interaction network model to predict the known binding sites, we used 18 different receptors. And for these 18 different receptors, we used 100 different crystal structures. Among these, 86 of them are ligand bound, either to orthosteric or allosteric, or both ligands. And my PhD student, Merve, did the statistical analysis. For this purpose, she constructed 4 data sets, experimental binding sites, their second shells, the theoretical binding site, and their second shells. For some cases, there were numerous crystal structures of the same GPCR containing different ligands with different binding sites. And in some cases, we had one crystal structure with one ligand only. And so this actually necessitated the use of the theoretical binding sites in this statistical analysis. And she collected all of these crystal structure and the ligand binding sites and assigned these as the experimental binding site data. Then she calculated the accuracy, precision, sensitivity, and specificity percentages. For the investigated structures, specificity and accuracy were high, and also sensitivity and the precision percentages improved with the consideration of the theoretical binding sites. So at the end, we concluded that the residue interaction network successfully predicts the non-binding sites of the investigated GPCRs. On the other hand, the residue interaction network model predicted other sites that can be considered as allosteric sites. Among the 18 GPCRs included in the statistical analysis, we concentrated on 8 of them to assess the dragability of the predicted sites using the site identification by ligand competitive saturation, or in brief, SILCs. All 6 calculations were done by Alex from the University of Maryland, and who is the co-founder of the design tech company 6Bio. My former PhD student Tuğçe did the residue interaction network calculations with Merve, and Tuğçe analyzed the results from the 6 calculations. As GPCRs are membrane proteins, the membrane protocol of 6 was employed, and the different types of solutes were overlaid in the system, and the oscillating chemical potential, grand canonical Monte Carlo, and ND simulations were performed using the charm force field. The probability distributions of the solutes were calculated over the trajectories using the Boltzmann distribution, and their grid-free energies were obtained where you are seeing is the frag maps. The frag maps were constructed showing the types of the solutes populated at different sites. 6 hotspots and residue interaction network were used to estimate together the location of the putative allosteric binding sites, where we evaluated the number of fragments and their averaged grid-free energies. To propose putative allosteric binding sites, we concentrated on the hub residues and the nearby hotspots, where we checked the number of fragments and their LGFE values. The consensus of both methods found the non-binding sites and also proposed other binding sites as drug targets. And these findings were recently published in ACS Omega. And among these GPCRs, we concentrated on the chemokine receptor CXCR4 that is overexpressed in many cancer types and is critical in HIV infection. And there exist several orthosteric and allosteric modulators available for the receptor for different purposes. Plerixafor, for example, is an FDA-approved drug, and it is a competitive orthosteric antagonist of CXCR4. And there are 10 compounds in clinical trials currently. And modulator binding sites are often located on the extracellular or transmembrane regions of the protein. And the benefits of allosteric drugs are well defined. So the CXCR4, we thought that this receptor may also accommodate intramembrane binding sites, as observed for many other GPCRs available in the GPCR database and protein data bank. This part of the study is published in the Journal of Physical Chemistry B, and we employed, along with the residue interaction network model, the Gaussian network model, to find hub residues, and then we employed 6 technology to assess the drugability of the predicted regions. As CXCR4 can be observed in monomeric and dimeric forms, we investigated both structures. First, we used the network models to identify the residues with a high ability to send and receive the allosteric signals, as well as to alter the conformational dynamics of the receptor. Among these residues, we also noted residues critical in function. Then we use 6 calculations to assess the predicted binding sites, and we propose 5 putative allosteric binding sites, 2 for the monomer and 3 for the dimer. And here, please note that the MA1 on the monomer is corresponding to DA2 on the dimer. And we employed 6 Monte Carlo for the to investigate these 5 putative allosteric sites, as well as the orthosteric sites for docking calculations based on the frag maps. FDA-approved compounds were investigated, and 20 compounds having high affinities for 1 or more binding sites were proposed. And we also checked out if these 20 compounds had a higher ability or not to the orthosteric sites, and these 20 compounds had a higher affinity for the proposed allosteric sites. Then George Lewis and Robin Flinko from University of Maryland did the in vitro assays to see if these compounds had an effect on the binding of the ligand CXCL12 and the monoclonal antibody 12G5. While the binding sites are close to each other, they are not the same sites, so we know that. And after the in vitro assays, we noted that actually all compounds affected the binding of the ligands. And interestingly, compound 20 enhanced the binding of CXCL12. Then they did a preliminary functional assay to understand if these compounds had agonist or antagonist activities. The assays were based on beta-arrestin recruitment and were performed in 1 concentration. For these assays, however, they did not note any significant effect for any of the hits. So they looked only at the beta-arrestin recruitment, and it is a possibility that the hit compounds might not affect the receptor dynamics without a direct, they may not affect the beta-arrestin recruitment. And further dose response studies actually are needed to see if these ligands have a real biological effect on the receptors. And we also applied the residue interaction network model to the bacterial ribosome, which is a protein RNA complex. As I mentioned, the model is not sensitive. It does not require any force field, so it can model any structure involving amino acids nucleosides. So bacterial ribosome is a great example for that. And there are many antibiotic targets in bacteria, but a large number of these are targeting this 2.5 megadalton machinery ribosome. And ribosome produces proteins according to the genetic code. It is composed of 2 subunits, a small subunit and a large subunit. And the small subunit is called 30S and the large one 50S in bacteria. And the small subunit accommodates the decoding center formed of 3 nucleotides, approving the cognate codon anticodon pairing. And many antibiotics bind near the decoding site. And messenger RNA binds the neck region of the small subunit. There exist 3 binding sites of transfer RNAs, bringing the amino acids to the complex. And these are called amino acid site, papillary site, and the exit site. Transfer RNAs with the amino acids enter the complex from the amino acid site and exit the complex from the exit site. The peptide bond synthesis is catalyzed at the peptidotransferase center, located on the large subunit, and the produced polypeptides leave the complex from a long exit ribosomal tunnel. And again, many antibiotics are targeting the peptidotransferase center and the entrance of the tunnel. So it is also known that the ribosome complex uses allostery to fulfill its function, for example, between the decoding center and the peptidotransferase center, or along the exit tunnel, etc. So exploring the allosteric mechanisms and the allosteric sites of this ribosome can lead to find new drug binding sites and developed or designed new compounds. These are particularly important because we are here in an era where antibiotics do not work. So the bacterial ribosome is at the center in this picture. So as the model is based on contact topology, we investigated 4 different conformations of the complex, which represent the elongation and termination steps of the translation process. We first focused on the centrality properties of the residue interaction network model, especially the betweens values. We constructed the distribution for the between scores and considered the top 5% quantile, and highlighted these on the ribosomal complex structure, where you see in magenta in these pictures. So, so when you look at these like they are forming certain paths between certain sites of the bacterial ribosome, and by definition, the nodes with high betweenness values have a high capacity to send and receive an information. So this may give clues about the allosteric mechanism of the ribosome. So, so we also determined that several intersubunit bridges, especially the 8, may serve as hubs in the structure, so they may be also evaluated as new drug binding sites. Here, actually, we concentrated on the bacterial ribosome in a TÜBİTAK project, and we investigated the functional sites using computational approaches such as molecular docking and molecular dynamic simulations. And 3 PhD students contributed to the work with master thesis students. And again, these we predicted some compounds, and these were tested by in vitro assays. And we found actually 4 inhibitors for the bacterial ribosome. And now we plan to do some structural assays using cryo-electron microscopy to see if these inhibitors actually bind to the locations where we predicted with the computational techniques. So I'm collaborating with Aylin Sungur and Aytan Karataş from Istanbul Technical University, and with Hasan Demirci from Koç University. So this is an ongoing project right now. And in another study, we investigated 3 allosteric enzymes of the glycolytic pathway. So this pathway and the related enzymes are highly conserved. Therefore, here we aim to find specific drug binding regions, especially for the S. aureus and some parasites. For this purpose, we use different computational techniques, and we employed residue interaction network model to predict the potential allosteric sites. We use the computational solvent mapping technique to assess the druggability of these sites. And we also use the elastic network model techniques to understand or to reveal the dynamic response upon ligand binding. And we a total of 15 crystal structures from SROs and 14 for the human were used. So I will be mentioning about the findings of these on these structures. I collaborated with Demet Aktan from Kadir Has University in this project, and my former PhD student Tuğçe was also contributed to the project. And for the phosphofructokinase, the first allosteric enzyme we investigated. So we combined the predictions of the computational techniques, and we look at the consensus sites. Here, the consensus sites are shown in cyan on the surface representation. And when a perturbation is made at the potential allosteric site, the dynamics of the catalytic region was noted to be restricted, which means ligand binding from the proposed site may have an allosteric effect on the enzyme. And for the human phosphofructokinase, no allosteric sites leading to this type of dynamics was found. So, we were again, I'm mentioning that we were concentrating on species-specific regions. And for the glyceraldehyde 3-phosphate dehydrogenase, or briefly GAPDH, we found a tunnel-like region as an allosteric site susceptible to perturbations, especially in SOs. Sorry. So we predicted that the targeting this tunnel region decreased the dynamics of the SO's GAPDH, whereas an increase in dynamics was observed for the human GAPDH. And we continued to investigate the allosteric mechanisms of this enzyme. We recently published a paper about that with my PhD student Zeynep and Demet Aktan. And also another one with Merve is under revision right now. So both papers are focusing on understanding the allosteric mechanism of GAPDH. And finally, SOR's pyruvate kinase. For this structure, predicted 3 different allosteric binding sites. One is located at the small interface. Actually, this is a known allosteric binding site with an inhibitor on it, so the structure is also resolved. And besides that, we also predicted the large interface and another site between the small interface and the active site of the enzyme. And so what we did with these predictions, first we collected all the data, all the predictions for these allosteric sites. We did drug screening, and we collected the mutual compounds for targeting or having affinity to these sites. And then we sent these to in vitro assays. And recently we published our results and reporting that we found 4 compounds with strong antimicrobial activity and 2 compounds with the complete inhibition of the pyruvate kinase. And in another study with, again, Merve, Demet Aktan, and Jet Sesal from Marmara University, we found 7, we predicted 7 modulators, allosteric modulators, and they were tested with in vitro assays. And now we are just preparing these for publication. So hopefully I was able to convince you that the residue interaction network model is a computationally efficient model, first of all, and it succeeds to map all known allosteric residues, functional residues, binding site residues on the same picture for all the investigated cases. And the only input it takes is a crystal structure. Actually, it is better to just work with multiple crystal structures for better predictions. And to assess the durability of the binding sites, we use different techniques. And among these, the SIX technology is a very powerful technique. And it uses fragment-based screening to evaluate the durability of the sites. And as it uses fragments, it is also possible to model a pharmacophore for de novo drug design. And just the combination of these techniques actually succeeded in predicting the non-binding sites, putative allosteric binding sites, as well as to identify allosteric modulators, which were all supported by in vitro assays. And the allosteric modulators can be evaluated with novel approaches, such as molecular glues or bitropic ligands or PROTACs, etc. So that was the end of my talk. I would like to thank my students, first of all. So, and as well as my current and previous collaborators who made possible this, the realization of this exciting work. And also I would like to thank you for attending this webinar, and I will be very happy to answer your questions.
Thank you for the precious talk, dear Dr. Ozge Hocam. Now we can continue with the questions. We can both take the questions as a written form from the chat part, and so I can read, or if you would like to talk, you can open your microphone. So, Büşra Özgüney asked, thank you for the fantastic talk. For some reason, my microphone is not working. Would be appreciated if Deniz can. Okay. My question is about whether residue interaction network model can be extended to represent an ensemble. I am particularly interested with the disordered proteins. The allostery in IDPs are a little controversial, but wanted to ask your thoughts.
Oh, it's a very good question. Thank you. So, here, in its current form, the residue interaction network model relies on the contact topology. So, which amino acids are interacting with which one is very important. So, we are using this information as the input. So, this, as it is, it will not work on the disordered proteins. However, we are currently using this model to harvest some information about the topology, three-dimensional, ordering of these amino acids with respect to each other. So by harnessing this information using machine learning, obviously, and AI, we can actually have something to say about the disordered proteins. But this is, this is an ongoing work, so we will test it if we are correct in our predictions or not. But it's a very good question. Thank you.
Is there any more question?
Thanks, Büşra.
So I think it is our end of our webinar session. Thank you for joining us today.
It was a pleasure for me.
And for us too. So I think we can end here. Thank you for again joining us today.
Thank you very much. Okay.
Ben kaydı
See you.
durduracağım hocam. İyi günler.